All right, well, hello. This is our first time presenting at Eyecelerator, and I appreciate the opportunity to get to share all of this with you. Our mission at Valitor is to develop long-acting ophthalmic drugs, and one of our key goals is to develop retinal medicines that only need to be administered 1 to 2 times a year. The technology we use to achieve this goal is called multivalent polymer conjugation. And what it enables us to do is take multiple copies of a variety of different drug moieties. We can conjugate those to long chain, single chains of hyaluronic acid. This is now creating a covalently clustered grouping of the drugs. And we use hyaluronic acid because it's biocompatible and endogenous, and it really maximizes the interaction that we can get with the matrix in the back of the eye that's really driving the durability for us. While we can work with a variety of different drug types, we're focused on large molecule drugs because we view that as being the greatest need in the space. I also want to make it clear this is not a slow release vehicle or a depot. These are covalently bound structures, and we can use the payload to define the specificity of the conjugate, and the biopolymer defines the PK properties. So I want to drill down on that a little bit because the idea of using biopolymers to increase size and durability is not necessarily new. Generally, you would take a single drug and put 1 or more copies of a biopolymer on it in order to get the durability that's needed. Although this has limitations because the more biopolymer you add, the more interference you get between the drug and its target. So at Valitor, we flip this around. We have a single chain of hyaluronic acid that we can make as long as we need to get the durability we want. And as long as we're increasing the copy number of that drug along the backbone, we don't have to worry about interference from the biopolymer. And in fact, we can set up multivalent interactions between the drug and the target in order to actually increase the potency on a molar basis. So this is the platform we're using for designing all of our drugs in the back of the eye. And our lead program, we call VLTR-559. It's a long-acting anti-VEGF biologic that is being developed for the treatment of wet AMD. In all of our in vitro studies, we see equivalent bioactivity to all of the licensed anti-VEGFs. And we've recently completed an in vivo study, an efficacy study based on a laser CNV model in transgenic mice that express human VEGF. And in this study, we were able to show that VLTR-559 has equivalent efficacy to inhibit that VEGF-induced neovascularization response in this model. So where we differentiate ourselves is on the basis of the PK properties. In a standard rabbit model, we're measuring half-lives for the VLTR-559 on the order of 12 to 15 days. And that's in comparison to more conventional drugs like Eylea that have a half-life closer to 4 days. So we're able to keep the drug in the eye and active in the eye for up to 3 to 4 times longer. And we can predict how that will impact the readministration frequency using this type of clinical efficacy model. So this is keeping track of the elimination of the drug over time after it's been administered. And based on all of the previous registration studies, we're able to estimate the percentage of patients that remain treatment-free as the drug's being eliminated from the eye. So I think this makes it clear with the longer elimination rate from the eye, we're getting a substantial increase in the duration of therapeutic efficacy relative to the marketed products. And we anticipate most, if not all, patients would be able to get to 6 months at least. And this sets up the opportunity for a really reliable retreatment protocol at once every 6 months with an anti-VEGF biologic. So our lead program, VLTR-559, is scheduled to go into the clinic towards the end of next year. We've been able to leverage the development of the hyaluronic acid backbone by putting other APIs onto that backbone in order to develop other pipeline products. And in fact, we're currently working with 2 industry collaborators who have APIs that need durability for differentiation in their space. And we've already now shown we can generate highly potent conjugates and that we're working towards license-enabling data. And from our perspective, there's no reason to stop there. As clinical mechanisms become validated, we can follow the established regulatory pathways and deliver long-acting versions of those drugs to the established patient populations that will need them. So as I mentioned, this is our first time at Eyecelerator. The timing is great because we are in a fundraising round. We're looking for investors who understand the value of these durable back of the eye and ophthalmic drugs. We're also here looking to meet with potential new collaborators who have APIs that need differentiation. If any of this sounds interesting to you, please come talk to me at one of the networking breaks. We also have 4 posters at ARVO, so there's plenty of opportunities for us to chat. Thank you very much.
Fantastic. Help me understand, first of all, excellent presentation, really exciting stuff for certain. Help me understand how does this, how would this be delivered in clinic, and safety? What do you anticipate in terms of that?
That's a great question. So I think one of the goals we had in developing the product was to ensure the experience from the clinician and patient's perspective was exactly the same as what they see with current anti-VEGF products. So one of the features of hyaluronic acid is that it shear thins, which means as you put it through narrower gauge needles, the viscosity drops. So we contemplate that this could easily be injected through a 30 gauge needle in the clinic. In our preclinical studies, we've gone down to 34 gauge needles, and it goes through just fine. And in terms of safety, again, this is engineered with safety in mind. We used hyaluronic acid because of its biophysical properties, but also it is endogenous to the vitreous matrix where we're injecting it, and hyaluronic acid has a long safety record in the back of the eye. So the polymer component has been largely de-risked from a safety perspective. We have done repeat dose non-human primate studies on a little bit of a different version of this drug, and it came through with a really clean profile.
That's great stuff. How big are these molecules all together? I mean, what sort of molecular weight? I know Kodiak had like 1 million million Dalton product. I assume these are smaller than that.
No, they're actually, from a molecular weight standpoint, longer, but they're linear chains. So one of the
less viscous,
complicating factors of the Kodiak molecules, it was a tendrimer, and that kind of branching molecule kind of increases the cross-sectional radius of the overall conjugate. Because we're linear, we actually thread through the tissues.
How long does it take to break down, and it just breaks into hyaluronic acid in the little...
Yeah, so the turnover rate of hyaluronic acid in the back of the eye is very long, so we anticipate it stays mainly intact while it's in the ocular tissues. Once it gets to the systemic compartments, as it's clearing out of the eye, there's a really high concentration of hyaluronidases, so we see really rapid breakdown in the systemic compartment, and the anti-VEGF antibody we're using is a single domain antibody, so it's cleared out through the kidneys also very rapidly.
I think it's really exciting. I guess something like pegcetacoplan that has a disulfide bond, I think things like that might be a problem though, because they could bind to themselves, the little moieties. Have you had any problem with drugs like that, or?
So it certainly is a challenge that we needed to overcome, but we actually have some background IP to get around that type of complication.
It's really exciting.
Yeah, just a quick question. One of the things that affects half-life in anti-VEGF drugs is the concentration 2, 4, 6, 8 lasts longer. So the 559, can you comment what's the concentration, milligrams per ml of anti-VEGF?
Yeah, so because we have a single domain antibody, it's much smaller than the existing anti-VEGF biologics. We really just have kind of the smallest, most stable unit we need for the anti-VEGF specificity. But we have looked at the currently licensed products, and we are targeting a molar concentration of the anti-VEGF units that are within the window of all the currently approved drugs.
Okay, thank you.
Fantastic. Thank you. Thank you, Bisley.
Great.
We're going to move on.